Have you ever wondered how scientists can peek inside a living brain and see which parts are busy at any given moment, almost like watching thoughts unfold? It is truly fascinating to imagine, and it relies on an incredible biological process. At its core, neurovascular coupling in functional magnetic resonance imaging, or fMRI, is the fundamental mechanism linking active brain cells, known as neurons, to changes in local blood flow. When neurons become more active, they demand more energy, and this demand triggers a localized increase in blood supply, which fMRI then detects as a change in the blood oxygenation level dependent or bold signal. This elegant process begins the moment neurons start firing more frequently. Increased neural activity requires a rapid supply of metabolic resources, primarily glucose and oxygen. Specialized support cells called astrocytes, which are intricately woven around neurons and blood vessels, play a critical role here. They sense the uptick in neuronal energy demand and releases signaling molecules like nitric oxide and prostaglandins. These molecules act as powerful vasodilators, meaning they cause the nearby tiny arteries, or arterioles, to relax and widen, dramatically increasing blood flow to that specific brain region. The surge in blood flow delivers an oversupply of oxygenated blood relative to the oxygen actually consumed by the active neurons. This creates a temporary imbalance where there is more oxygenated hemoglobin and less deoxygenated hemoglobin in the local capillaries and venules. Oxygenated hemoglobin has different magnetic properties than deoxygenated hemoglobin. This difference in magnetic susceptibility is precisely what fMRI scanners are designed to measure, providing an indirect but powerful window into brain activity. Understanding this cascade is vital for interpreting fMRI data. The fMRI signal itself is not a direct measurement of neural electrical activity, but rather a reflection of this metabolic and hemodynamic response. Researchers use this principle to create detailed maps of brain function, localizing areas responsible for tasks like language processing, memory recall, or motor control. It is also important to note that various factors can influence neurovascular coupling, including age, certain medical conditions, and even medication, which can sometimes lead to what is known as neurovascular uncoupling, where the blood flow response does not accurately reflect neural activity. A key concept in this field is the hemodynamic response function, or HRF. This is a characteristic model of the blood flow changes over time following a brief neural event, typically peaking several seconds after the initial neuronal firing. It serves as a blueprint for how the brain responds metabolically. While fMRI provides remarkable spatial resolution, its temporal resolution is limited by this inherent delay and the relatively slow nature of the blood flow response. The bold signal also represents an aggregate of activity from millions of neurons, not individual cellular events. This gives us a high-level view of brain function, revealing widespread patterns rather than fine-grained neural codes. Ultimately, neurovascular coupling is the sophisticated biological link that transforms the brain's electrical chatter into a measurable signal of increased blood flow. By harnessing this intricate dance between neurons and blood vessels, fMRI has revolutionized our ability to study the living human brain, allowing us to explore the neural underpinnings of cognition, emotion, and behavior, and advancing our understanding of both healthy brain function and neurological disorders.